the first session. So I'm happy to have Anthony from University of Michigan to present his work on ontological representation, modeling, and analysis of parasite vaccine. This is an expansion to Vaccine Ontology. Anthony, take it away, please.
Hi, and thank you for letting me skip my first slide. So before we begin, I want to establish that for this talk, we're mostly focused on 3 ontologies and knowledge bases. The first is vaccine ontology, which is an OBO focused ontology on the focused on the representation of vaccines, adjuvants, antigens, and all pathogens, anything that a vaccine interacts with. This ontology is used to help inform the Vaccine Investigation and Online Information Network, which is a primary knowledge base that we use to store information on different vaccines. I would also like to mention that as part of this work, we collaborated with the ontology of parasite life cycles, mainly because we wanted to import various terms related to parasite life cycles from this other OBO aligned ontology. So the main reason for this collaboration is that we are interested in this idea of pathogenic parasites. So a parasite organism by itself is any organism that engages in parasitism of another organism. That is, it's drawing some nutrients or causing some sort of negative relationship as it benefits from at the cost of another. So in this context, we're mostly focusing on parasites that can cause a disease process in their host species. So malaria, leishmaniasis, Chagas disease are examples of parasite-based diseases that affect humans. But we also have diseases such as malaria affecting poultry, and neospora affecting cattle, as other parasites which affect livestock. So there are 3 major categories of pathogenic parasites, which are protozoans, helminths, and ectoparasites. For this talk, I'm mostly going to be focusing on 2 specific clades of protozoan parasites, which are apicomplexans and mastigophorans. Both of these are single-celled organisms that have unique organelles, which is why they're classified as such. And they're mostly interested because of their response in causing a disease process in humans or other key livestock. So the main motivation as part of this study is that for the longest time, parasite vaccines have been slow to develop, simply due to the actual understanding of what makes a good antigen that is needed to form a good vaccine candidate is not something that's really well known. Because of that, we've been mostly focusing our updates on violin, on virus and bacteria. So we have this gap of a couple different years before I joined the lab where we did not really have anyone focusing on updating parasite vaccines. And with the recent updates in the last few years, this seems like a good time to update that. However, when we are looking at some of the new parasite vaccines, we notice that many of them are targeting specific life cycle stages for the parasite pathogen instead of focusing on the entire microbial life cycle. So normally VO assumes that whatever vaccine we have will be effective against the pathogen, end of story. So it doesn't matter if it's a specific life stage or specific strain or other variant. If we have a vaccine that works against a species, it will always work on that species. And since we're seeing that, hey, some of these parasite vaccines are targeting specific life stages, we wanted to take a look and see that, hey, do all the parasite vaccines we have affect all of the life stages, or is it just the fact that, hey, this study says it only affects the sporozoite life stage, that it only affects that specific one? Additionally, we're hoping that with the collection of further vaccine data, especially for parasites, we can start to find some common features within these parasite antigens in order to further aid further R&D and better understanding of what makes a good vaccine for parasites. So for the workflow, it's relatively simple. We had a bunch of undergrad students and go through PubMed or clinicaltrials.gov looking for any information on new parasite vaccine studies that include some immunological response or some immune efficacy for what's going on. From these trials, we added the data into Violin as part of the data collection. And then we also take a note for the different parasite life stages that were mentioned as the target for the actual pathogen, or also the actual gene IDs that are listed. So once we've collected all this information, we generated new ontology terms into either OPL or into vaccine ontology directly using the tools listed on the arrows. From there, we imported some of the terms from OPL using Ontofox, and that's what we used after coming up with our actual modeling. We had a second one where we wanted to take a look at patterns for the actual gene sets that we found. So after collecting all the vaccine collection, we took a look at any of the NCBI gene IDs we could, and put that into David, and then performed the gene set analysis. So for the collection and annotation into Violin, we have the entry showing the 10, all of the different parasite vaccines with at least 10 vaccines showing up. So right now, for all the pathogens, we combine them for the actual diseases that are associated with, since I figure most of the audience would recognize the disease more than the actual scientific name for these parasites. Altogether, we have 260 parasite vaccines within Violin, which was an increase from the 77 that we started with initially. From there, we are covering 20 different parasite species, and we have found a total of 198 unique antigens that are across these different species. One surprising factor we did find was that only the vast majority of the vaccines that have been in development were focused on humans, with only 12 of the vaccines targeting non-humans, typically either cattle or poultry, and most of these were from entries that we had before the update. So when we take a look at the different protozoan parasite life cycle, we have this nice example of Plasmodium falciparum. So in each of the colored boxes over here, we have the different parasite life stages, which are showing next to the actual host stage that the parasite cell infects. So we can see that for Plasmodium, we affect the human liver, we affect blood cells as part of the human blood stage and the erythrocytic cycle, which is responsible for the malaria process, before moving over to the mosquito stage. And while each of the boxes contains a list of vaccines that actually target this stage and the number of antigens that are utilized, I would like to direct your attention to the red box in the mosquito stage. So out of the 30 or so Plasmodium falciparum vaccines that are shown on this figure, only 3 of them are listed as affecting all stages, which meant that our previous model was not as effective in capturing the actual reality of parasite vaccines. This is especially true as some of the antigens that are actually expressed will be unique to specific life stages, which means that our primary justification for using an antigen, that is, it will have the host generate an immune response when it recognizes that antigen, will only occur for that life stage, which means that many of these are limited on where you can apply the vaccine and still have it to be effective. So as such, we needed to establish a new pattern to help describe this behavior into vaccine ontology. So fortunately, the change for this was relatively simple, which is that the general pattern is that when we go to the immunizers against microbilation, we specify the specific parasite organism instead of the organism. And in this concept of parasite organism, we're saying that the organism, when it participates in a specific parasite life stage. So that would be the example of the sporozoite, the organites, or something similar. So by doing this, and by enlisting the actual vaccine antigen for when it persists in the life stage, we're able to cover this more limited window of opportunity for the vaccine to be effective. So if we were to look at a practical example using the cell-toss vaccine, we can see that the Plasmodium falciparum PF cell-toss protein vaccine utilizes the cell-toss protein, which is found in two different Plasmodium life stages, that of the sporozoite and that of the okinetius. So from this situation, we can say that, hey, we have experimental evidence that the cell-toss protein vaccine works against the sporozoite protein, and we also know that it utilizes the cell-toss antigen. However, we can also, by listing the antigen as an additional parasite life stage, we can provide an additional potential coverage or location for where it can intersect and have some sort of efficacy. So if we were to go into some of the older vaccines when it comes to update, we have for the Mascarix. This mainly means that the only thing we have to change for some of these older entries is that we just update immunizers against microbilation to specify against a specific life stage. In this case, the Plasmodium falciparum sporozoa. However, there is one other pattern that we found for something called a transmission blocking vaccine. So we define a transmission blocking vaccine as any vaccine that has a relationship that blocks transmission of pathogen by a vaccine, which is an additional relationship where we have a vaccine and a parasite, such that the vaccine has an additional effect that it inhibits further transmission of the parasite among additional host of vectors. That is beyond the initial target. So right now we list this as a subproperty of both the primary immunizers against microbilation, but also of the capable of blocking transmission of life cycle property, which specifies a further relationship downstream. So what this looks like with the example of VDOS2023 is that we need 3 axioms to fully cover a transmission blocking vaccine. The first is, of course, the definition of blocks transmission of pathogen via vaccine against some pathogen, in this case, Plasmodium falciparum. We still have the relationship of immunizers host some Homo sapiens, so this is a vaccine that you target and inject into humans. From there, we have a third property, which is vaccine immunity response transferable to organisms, some anopheles. So anopheles are mosquitoes, and this refers to a specific property where that the immune response generated by the vaccine for humans against Plasmodium is intaked by the mosquitoes as a means to which will then inhibit the development of further Plasmodium falciparum within the midgut of the Anopheles mosquito. So in this case, we have 3 primary focuses, which is we have the pathogen, the host, and then a secondary host or vector, which will receive the immune response generated by the first host in order to inhibit the response of the target pathogen. So altogether, when I was looking over the paper, we added about 417 newtons into VL. This number is out of date since we've made some corrections and further updates between when we submitted the paper and for the conference and later on. So most of these new terms are related to either parasite vaccines or parasite antigens, where we had a significant tripling of the number of parasite vaccines and a doubling for the total number of antigens included in VL. We've included 15 new parasite vaccine categories. Right now, this is mostly occurring for specific ancestors for some specific species, instead of just using the general genus name for some of the vaccines. We've also added 5 new vaccine relationships, almost all of them relating to the transmission blocking vaccine. We have also imported 107 new terms from the ontology of parasite life cycles, which we use as the actual target as part of the microbe and as part of the life stage story. So once we've made all these updates on the ontology, I also want to focus on the use case, which is trying to identify some common features found within these parasite antigens. So after mapping the different protective antigens that we've included, we ran into a slight snag in that some of them weren't recognized as having valid gene IDs, which meant that for our next part of the analysis, we went down from 198 protective antigens, and we have that being reduced to 140 gene IDs. So following that information, we performed gene set enrichment analysis using David, which will provide us for a given list of genes in a specific organism, some common ontology annotations using either Gene Ontology, Unicode, KEGG pathway for the source of these annotations. Now, ideally, this methodology can also be applied to identify transcriptomic data for different parasites in different life stages, which we could use to better understand some of these patterns later on or for other interactions. But as part of the initial analysis, we were purely focused on just the vaccine antigens that we have seen for the list. So, which meant that with the reduced gene list, we really got significant terms for 3 major species: Toxoplasma gondii, Plasmodium falciparum, and Trypanosoma cruzi. I've included the disease name in parentheses, along with whether or not it's an apicomplexan or a masticaform, so the type of protozoan in parentheses. So while we had multiple terms, I mostly focused on the highest, the most significant term within a cluster that we found using David. So for the apicomplexans, you can see in blue that we had some terms related to the actual disease process, which was still a smaller fraction than we expected. It was still showing up as significant for some of the process. We also noticed that some of the red boxes that we highlighted are properties that we found are related to specific localization or functions for some of these proteins. So for the toxoplasmosis protein, we see that we have ontology annotation terms relating to protein kinases, which tend to be internal to the actual cell. Meanwhile, when we take a look at the Plasmodium parasite, we tend to find localization occurring for these antigens occurring within the plasma membrane or on the extracellular side. For the T. cruzi, we found something with the multicilium, which is the unique feature of masticaforms. But so while we're starting to see some differences for what's going on, this is still a very small sample size and is just used as an initial baseline to show that, yes, we're starting to be able to predict some significant features for these antigens. And this is something that we want to investigate further as part of our future directions. So right now, this is just the master list for other antigens. And that was partially done because if we were to restrict these vaccines further to unique life stages within the actual parasite, we're going to start dealing with the issue of the gene list being too small. But one thing that we do want to investigate is using the antigen list, take a look at the transcriptomic expression at the different life stages to make sense of what's going on. A secondary focus that we want, at least on the ontology side, is whether or not we should do a similar update for viruses. So as part of the coronavirus infectious disease ontology, we started including information on different notable variants of concern or variants of interest, which represent strains that have diverged enough to have at least enough of a change in the immunological profile needed for either vaccination or for can reaffect a populace. So one point of future discussion is whether something similar to this model should be done to update some viruses. We also had some discussion with bacteria on whether or not for representation for transformed, but that one is a bit more inconclusive. So continue with that. We want to continue with using this as a use case. We want to use some of the gene enrichment set analysis for reverse vaccinology analysis. This is more of a future direction for mine, which is incorporating some of the life stage pattern for prediction of further parasite antigens that would be good quality. And we also want to follow up and see if we can find further patterns, if we can expand this out to other apicomplexans or masticaform parasites. A third category, and this is for violin, is just searching for additional vaccines. So our study focused on species we know had parasite vaccines already successfully developed or in development, but that still leaves a large category of other parasites that we can potentially include as part of the actual research. So that includes some additional protozoans, but it also includes some intestinal worms and also fleas and other mites. We haven't really investigated to see what vaccines are out there, but that's a potential future that we want to investigate further in the coming year. Right. Thank you. I tried to speed that up. So are there any questions?
Great, thank you, Anthony. Are there any questions from the audience?
Kevin Burns will ask one. Do you have any planned interaction with IEDB, who's mapping all the antibody to antigen interactions?
As part of the directly related to the Violin VO update, or as part of future directions for RV?
Okay, whatever works.
So that is something that I want to follow up as part of my thesis work, through either on if there's greater predictive factors for the IEDB website, either directly or either as a additional analysis that can be performed after RV design. So currently Violin hosts Vaccine and Vaccine ML, which are different web tools, which in addition to predicting or providing some information against predictive antigens is hooked up to look for IEDB antigen interactions. But currently for the specific epitopes, that's not something that we plan on including directly into either VO or OPL. So it's something I'm looking towards, but it's not directly related to the ontology side. Does that answer your question?
Yeah, just general curiosity. Yeah. If I can follow up briefly, so I'm working on related efforts, but one of the challenges is how do you characterize an antigen? And if you said that, I may have missed it, and I'm sorry. But it's tricky because it runs from it's basically a molecule, right? It runs from this subdomain or this chemical motif of this protein and so forth. And so coming up with a vocabulary for that is tricky.
Yes. Admittedly, we take the very simple one of if you have experimental evidence that it works as an antigen, it's an antigen. So that's what we use as our basis. RV, when it comes to prediction of antigens, is more speculative, but that's unrestricted to any valid peptide sequence or protein sequence, which then can be predicted. But again, anything that we're currently predicting in silico as an antigen is not going to be added to the database or VO. That's just a tool to help scientists hopefully run in experiments to then confirm that, yeah, this is an antigen.
Great, Anthony, quick question. You might have already mentioned, so because of the complex, life cycle, I wonder if people are actually using kind of combinatorial approach in combining multiple, vaccines together in malaria.
So that is an interesting point. From what I recall with malaria, all these different vaccines are being researched or separately in terms of efficacy, and either combined with research for anti-malarial drugs to slow down transmission, or focusing more on few life cycles. While I do think that could be a good potential option, I think right now most of the research and work is done as, okay, if we vaccinate this specific life stage, it will have an effect on reduction. While the other issue is that the Mysterix vaccine that I mentioned, that is, to my knowledge, the only vaccine that is currently licensed for use against malaria itself. So such combinatorial approach would probably wait until you get other vaccines officially licensed, and then you can do a separate study to see how it goes. So
yeah,
again, effective parasite vaccines, especially for things that are endemic to human populations, is just really recently starting to gain steam and something effective, because Mysterix took like 30 years of development in order to get something. So.
Okay. All right, great. Thank you. Are there any questions from the audience? If not, let's thank the speaker, and we can move on to the next one.